Parkinson's disease is a condition affecting dopaminergic cells in the brain, specifically in the substantia nigra. These cells are responsible for producing dopamine, a key chemical messenger involved in voluntary movement in the brain. Understanding the pathology of this disease is key to developing effective treatments in patients. But how did we discover dopamine's important role in the brain? For that, we must turn to the discoveries of Dr. Arvid Carlsson. Arvid Carlsson was a Swedish researcher best known for his discovery of dopamine and its important implications in Parkinson's disease.
Arvid Carlsson was born in Uppsala, Sweden in 1923 and is best known for his work on the neurotransmitter dopamine and its effects on Parkinson's disease. He entered medical school in 1941. His education would become interrupted due to his military service obligations in Sweden. He would later finish his degree in 1951 and became a professor at the University of Lund and would move to Göteborg University in 1959. His main research started in 1957. Dr. Carlson would conduct his experiment that showed that dopamine was a neurotransmitter in the brain. He would also work on developing an assay to measure dopamine in the brain. Further investigations by Dr. Carlson showed that L-DOPA, one of the precursors of dopamine, was an effective treatment for the symptoms of Parkinsonism and is still used today for the treatment of this disease. Due to the contributions in the field of medicine and physiology, he would later be awarded the Nobel Prize for Medicine and Physiology in 2000 due to his contributions towards the research of the neurotransmitter dopamine.
Dr. Carlson's breakthrough discovery came when he was investigating the effect that reserpine has when administered to mice and rabbits. This drug causes paralysis and ptosis of the eyelid when given in large doses. It is known that reserpine blocks the reuptake of monoamine neurotransmitters, which include serotonin, dopamine, norepinephrine, and epinephrine, thus depleting their levels in the brain. However, the differential effects between these neurotransmitters were poorly understood. Dr. Carlson showed that administration of L-DOPA, a dopamine precursor, to a reserpine tranquilized mouse or rabbit will result in a temporary return to normal movement in these animals.
So, in order to see if depletion could be the cause of the behavioral changes induced by reserpine, we gave DOPA. And on the top here we have reserpine-treated rabbits, and the one underneath are the same rabbits 10 minutes after an IV injection of DOPA. And so we were really, just like the rabbits, extremely excited by this finding.
This study laid the groundwork for providing evidence that dopamine is not only its own distinct neurotransmitter, but also that it plays an important role in voluntary movement.
In the early to mid 20th century, it was widely believed that 3-hydroxytyramine, more colloquially known as dopamine, was simply a precursor to both adrenaline and noradrenaline. However, in the late 1950s, Arvid Carlsson's experiments led him to presume that dopamine was not simply a precursor, but its own independent molecule with intrinsic functions.
We also found dopamine occurs normally in the brain in amounts that are at least as high as those of noradrenaline, arguing against its role as merely a precursor.
Via a modified fluorometric assay, Carlsson and his colleagues were able to once and for all successfully display the fluorescent distribution of dopamine in certain areas of the brain. Previously, fluorometric assays were unable to distinguish between molecules like noradrenaline, dopamine, and adrenaline. However, Carlsson's experiment modified the pH to around 5 using acetic acid in order to obtain a strong fluorescence for dopamine itself, while also excluding the fluorescence of both adrenaline and noradrenaline. Confirmation of this stems from the relatively shorter wavelengths of fluorescence observed after pH adjustment, values ranging between 345 and 410 nanometers, compared to adrenaline and noradrenaline, which have comparatively larger observed wavelengths. Further investigation via ion exchange chromatography revealed that the concentration of dopamine in the brain of experimental rabbits is similar to that of noradrenaline. As a result of these findings, Carlsson and his colleagues for the first time theorized that dopamine may not simply be a precursor to noradrenaline, Rather, dopamine is its own independent neurotransmitter.
So this seemed to us to be an extremely exciting finding. We felt like maybe we had kind of a Rosetta Stone here. We could have an entrance to the chemical and access to the chemical language of the brain by this finding.
Dr. Carlsson won a share of the Nobel Prize in Physiology or Medicine in 2000 for his discovery of L-DOPA. L-DOPA is still the primary treatment of motor symptoms in Parkinson's disease. L-DOPA is converted to dopamine by the enzyme dopa decarboxylase. However, when dopamine binds to peripheral tissue receptors, it can cause the following side effects such as nausea, vomiting, and hypotension. Furthermore, dopamine cannot exit the peripheral tissues and cross the blood-brain barrier due to its chemical structure. The clinical solution to this is to administer L-DOPA in combination with another drug known as carbidopa. This is because carbidopa is not capable of crossing the blood-brain barrier like L-DOPA. It instead inhibits the enzyme dopa decarboxylase mentioned earlier in the peripheral tissues. Therefore, by inhibiting this enzyme in the peripheral tissues, this allows L-DOPA to remain as L-DOPA. It then is converted into dopamine only after it crosses the blood-brain barrier. This extends L-DOPA's half-life and its availability to the brain where it is needed. This reduction in peripheral conversion also reduces the side effects mentioned earlier. In conclusion, Dr. Carlsson's discovery of L-DOPA was a breakthrough in neuroscience and is still extremely relevant to the treatment of Parkinson's patients today.